Hi everyone, I'm Danny, and Rachel, Josh, and I will be tracing the development of the drug class of beta blockers, which have been called one of the most important contributions to clinical medicine and pharmacology in the 20th century. While not the disease they were initially developed to target, beta blockers have come to be one of the most important types of drugs used in the treatment of hypertension, also known as high blood pressure. Hypertension is one of the most common diseases in the world, affecting over 1 billion people. According to data from the World Health Organization, hypertension is prevalent at similar rates throughout the globe, primarily affecting lower income to upper middle income countries. Hypertension is defined as systolic blood pressure of 130 millimeters of mercury or higher, and or a diastolic blood pressure of 80 or higher. This increased pressure poses a serious health risk, making hypertension a silent killer. Normal blood pressure is crucial for ensuring proper organ function, but hypertension can cause damage to the endothelial cells that line the inside of blood vessels. This damage can lead to myocardial infarction, heart failure, stroke, or kidney failure.
In order to regulate hypertension, beta blockers target beta adrenergic receptors. There are three types of beta adrenergic receptors classified as subtypes beta 1, beta 2, and beta 3. Beta blockers can differ in their selectivity for each receptor subtype. Non-selective beta blockers bind both beta 1 and beta 2 adrenergic receptors, while selective beta blockers typically target just beta 1 receptors. Structurally, all of the beta receptors are similar. The ligand binding site is formed by 7 membrane-spanning domains. Aspartic acid 113 in transmembrane domain 3 is the most important residue. Aspartic acid 113 is conserved in all adrenergic receptors, and its carboxylate is believed to stabilize the amine of the ligand through hydrogen bonding. In order to bind the beta adrenergic receptors, beta blockers must follow a certain pharmacophore. They must contain an aromatic ring and a beta ethynylamine. The binding site of the beta adrenergic receptor is the same for beta blockers as for the endogenous ligand. Notably, the binding site is built on hydrogen bonds and not based on covalent bonds. Both the hydroxyl group and amine in the pharmacophore can participate in hydrogen bonds, as well as different atoms placed in the variable regions for a potential drug. Beta 1 and beta 2 adrenergic receptors are located on the heart and are activated by catecholamines, more commonly known as norepinephrine and epinephrine. When norepinephrine binds the beta 1 and beta 2 adrenergic receptors, it activates the CAMP signaling cascade. As a result of the cascade, a rise in calcium levels causes increased heart contractility and heart rate, both contributing to an increase in blood pressure. Beta blockers act to competitively inhibit the beta adrenergic receptors. If a beta blocker binds the receptor to prevent the binding of the endogenous ligand, it inhibits the normal effects that act through those receptors. A decreased effect would lessen heart contractility, reduce cardiac output, and ultimately reduce blood pressure.
The history of beta blockers began with the surprising discovery of dichloroisoprenolone, or DCI, by the Lilly Research Lab in 1958. These researchers were screening chemical derivatives of isoprenolone in order to develop a long-acting bronchodilator compound. Isoprenolone is an analog of adrenaline that selectively binds to beta adrenoreceptors and is able to induce dilation of the bronchial passages as one of its effects. During the screening process, the researchers discovered that tracheal tissue lost its ability to relax to subsequent adrenaline exposure when DCI was administered, prompting them to classify it as a beta antagonist. This study caught the attention of James Black, who was trying to find a way to selectively inhibit binding to the beta adrenoreceptor. However, Black's group found that DCI blocked the stimulative action of isoprenolone in some tissues while displaying agonist activity in others. Thus, they classified it as a partial agonist. Unlike a full agonist, which is able to elicit a complete response like the endogenous ligand, a partial agonist induces a submaximal response when bound to the receptor. In the case of beta blockers, this submaximal response is referred to as intrinsic sympathomimetic activity, indicating the production of some background sympathetic response when bound to the beta receptor. Additionally, these partial agonists display antagonistic properties as they are able to completely inhibit the actions of more effective agonists via high affinity binding. Ultimately, Black wanted to find a way to modify DCI to develop a lead that retained specificity for the beta adrenoreceptor while also completely inhibiting receptor activity. Since the modification from isoprenaline to DCI resulted in weakened agonist activity, Black decided to extrapolate the structural reasons behind this reduced activity in order to guide the optimization process. He reasoned that substituting the hydroxy groups of isoprenaline with chlorine atoms caused the size of the side groups to increase and for them to become conformationally fixed due to pi bonding. Thus, he proposed making a naphthyl analog of isoprenaline in which the chlorine groups were substituted by a fused benzene ring, as this modification would further increase the size of the side groups while maintaining conformational stability. As a result, Black synthesized pronetholol, which fulfilled his goal of developing a beta antagonist without any agonist activity. Looking at the beta adrenoreceptor binding pocket, there are several interactions that stabilize DCI and pronetholol binding. For instance, both compounds rely on hydrogen bonding between their secondary alcohol and amine groups and the asparagine 312 and aspartate 113 residues. Regarding the phenyl substituent, the chlorine and naphthyl side groups enable hydrophobic interactions with the TM5 and TM6 regions of the binding pocket, such as phenylalanine 290 and tyrosine 199. Furthermore, as hypothesized by Black, the increased size of the naphthyl side group compared with the chlorine groups allow for stronger dispersion forces to facilitate high affinity binding between pronetholol and the TM5 and TM6 regions.
Pronetholol was described to have side effects including disorientation, nausea, and vomiting. More importantly, pronetholol was shown to be a carcinogen in mice, so scientists set out to discover a less toxic beta blocker. In 1964, Dr. James Black discovered propranolol, a non-selective beta blocker. The structure of propranolol differs slightly from the structure of pronetholol in that it has an additional oxygen between the aromatic rings and the carbon containing the alcohol group. Aside from having less toxic side effects, propranolol was shown to be about 10 times more active than pronetholol in antagonizing increases in heart rate. After only 2.5 years of clinical trials, propranolol was released under the brand name Inderal for the treatment of angina and arrhythmias. Since then, propranolol has been approved for oral use in patients with systemic hypertension. Taking a look at the pharmacokinetic properties of propranolol, the molecule satisfies all of Lipinski's rules. It has a molecular weight of 259.34 grams per mole, an experimental log P of 3.48, 3 hydrogen bond acceptors highlighted in red, and 2 hydrogen bond donors highlighted in orange. Additionally, propranolol satisfies the size restrictions determined by Guida because it only contains 19 heavy atoms. Compared to other beta blockers, propranolol is highly lipophilic due to its low capacity to form hydrogen bonds. For propranolol, the distribution ratio comparing N-octanol to water was 20.2, a value that is 1,000 times larger than the distribution coefficient of other beta blockers. When comparing the structure of propranolol to other hydrophilic beta blockers, such as sotalol, you see that the addition of a hydrophilic group capable of making multiple hydrogen bonds makes a large difference in terms of lipophilicity. Because of its lipid-soluble properties, propranolol has an absorption rate of greater than 90%. That being said, propranolol's lipophilicity also subjects the molecule to a large first-pass effect, which ultimately decreases the overall bioavailability of propranolol to about 30 to 40% dependent on initial dose. During propranolol's first pass to the liver, about 70% of the drug is metabolized. However, the main metabolites of propranolol are pharmaceutically active. The drug produces two main metabolites. First, propranolol can undergo conjugation with glucuronic acid at its hydroxyl site to form propranolol glucuronide. Second, the aromatic ring of propranolol can be oxidized to form 4-hydroxypropranolol. This metabolite has the same amount of beta-blocking activity as its parent molecule. Volume of distribution varies greatly among beta blockers. The volume of distribution of propranolol ranges from 3.3 to 5.5 liters per kilogram. The lipid-soluble drugs like propranolol appear to gain access to the central nervous system more easily and rapidly than other beta blockers, which could explain why certain drugs like propranolol are more likely to cause centrally mediated side effects such as vivid dream. The figure shown illustrates the correlation between clearance routes and lipophilicity of a beta blocker. As the figure shows, the lipid-soluble beta blockers like propranolol are cleared almost entirely through the liver, whereas hydrophilic drugs toward the right of the figure depend on renal clearance. More explicitly, to illustrate this point, the hepatic clearance rate of propranolol is approximately 1,000 milliliters per minute, and its renal clearance is about 0 milliliters per minute. Due to its rapid hepatic clearance, the half-life of propranolol is relatively short at around 3 to 4 hours. However, in order to be more manageable for patient use, the drug company behind Inderal developed a sustained-release formulation of the drug that maintains plasma concentrations of propranolol within the therapeutic window for 8 to 11 hours. With this modification in drug delivery, propranolol can be taken once daily for hypertension patients.
The drug industry is, first and foremost, an industry, and this is demonstrated in the development of atenolol, the first selective beta blocker. In the midst of the commercial successes of propranolol, it was eventually reported that propranolol possessed local anesthetic properties, raising questions about the actual efficacy of the drug. Even before then, though, the company that made propranolol, Imperial Chemical Industries, or ICI, was facing competition in the beta blocker market. As one member of ICI stated at the time, quote, "We must energetically use our shrinking scientific lead to maintain our commercial lead." The local anesthetic properties were not the only concern that existed regarding beta blockers. There were also worries about some of the side effects already mentioned about propranolol. Research and development eventually resulted in the molecule Practilol in 1968. By coincidence, the researchers found that Practilol blocked the beta-1 receptors in the heart, but not the beta-2 receptors in bronchial or vascular smooth muscle. This selectivity was a big deal for beta blockers, because propranolol's lack of selectivity was a concern for individuals with pre-existing conditions, such as diabetes or asthma. However, Practilol was not as potent as propranolol. Practilol was eventually withdrawn from the market too in 1975, after it was found that its use led to serious side effects like blindness. In order to maintain market dominance then, ICI set out to find a drug that 1. Was more potent, 2. Had no agonist activity, 3. Was cardioselective, 4. Was free of local anesthetic activity, and lastly 5. Was very hydrophilic. In effect, the researchers were trying to combine the best parts of these two previous drugs into one ideal beta blocker. Making a molecule about as potent as propranolol was not difficult, so, with the selective optimization of site activity, or SOSA, approach, the researchers first considered cardioselectivity in an effort to make the first major drug in the class that was selective. It was hypothesized that Practilol's selectivity was due to the acidic hydrogen on the amine attached to the benzene ring. With some modifications in order to make the molecule easier to synthesize, the researchers eventually developed atenolol. The new side chain the researchers added made the drug less difficult to synthesize and cardioselective. The researchers were luckier than they could have imagined initially, though. With no other modifications needed, atenolol also already had no partial agonist activity, no anesthetic activity, which would ensure that the drug's effects were the result of relieving hypertension, and it was very hydrophilic. All 5 of the researchers' initial goals were met. Obviously, atenolol was not the only molecule studied during this drug development process, and so this story also presents the way in which drug researchers must prioritize certain aspects of drug development, While atenolol was the most potent selective molecule developed, it was actually not more potent than propranolol, but other aspects of the drug, including the lack of serious side effects like in propranolol, cardioselectivity, and being longer lasting, made atenolol a promising drug, even though potency was sacrificed. A number of pharmacokinetic properties about atenolol made it very unique at the time of its development for beta blockers. The primary factor is lipophilicity, with an experimental log P of 0.16, vastly different from propranolol's log P of 3.48, and thanks to its polarity, atenolol is one of the most hydrophilic beta blockers today. This hydrophilicity directly affects the ADME properties of the drug. Atenolol is rapidly, although incompletely absorbed. As can be seen in the table, multiple studies found that the drug's oral bioavailability to be between 50 and 60% compared to an IV bioavailability of around 90%. This can be attributed to the fact that because it is so hydrophilic, atenolol cannot easily penetrate the cell membranes, especially in the intestine. Due to this inability to penetrate cell membranes and be easily absorbed, atenolol is minimally metabolized by the liver. This lack of metabolizing can be seen in the very high urinary recovery percentages. Nearly 50% of the unmetabolized drug can be recovered in the urine after 24 hours. The reduced absorption that results from the hydrophilicity of the drug also affects the half-life of the drug. Because renal elimination is the primary route taken by the drug, atenolol has a half-life of about 6 to 9 hours.
We will conclude by using recent sales data from 2007 to give a sense of the types of beta blockers that remain prominent today. Toprol XL, which is the AstraZeneca formulation of metoprolol, is the top-selling beta blocker by a substantial margin due to its convenient 24-hour length of action in the body, allowing for it to be taken once a day. Additionally, propranolol, indarol, and atenolol remain popular beta blockers that are still often used.